Okay, so thank you everybody for joining our webinar today. This webinar is going to focus on lessons learned and a performance review from the multiple geological settings from the applications of PlumeStop that have taken place to date. It is actually the third webinar in the series of webinars that we've had on PlumeStop. The earlier two are available for download from our website and can be reviewed at your leisure. To recap, the first of these really dealt with the core concepts of PlumeStop and the underpinning science behind it. How did we know it did what it did? What are the key performance factors that were important in the laboratory and in the field of showing how it would work, etc. The second webinar concerned more with field application, the transport of the reagent through soil and through mixed permeability systems, the bioregeneration, the coupling of the sorption with the degradation, etc., and presented very many case studies from that point in time of the use of the technology. This is the third webinar in the series, and our emphasis of this is going to be slightly different. We will naturally deal with a short recap of the technology basics and a bit of background. I won't go through the full content of the first and second webinar, of course, but I'll provide enough information to really frame the main part of this webinar. The webinar will concern usage statistics of all of the sites that we have undertaken to date, and will provide some aggregate field performance to provide an indication of how reliable the technology has been so far in practice. I will then also deal with some of the lessons learned from these applications. I'll then spend some time towards the end of the webinar looking at the road ahead, some of the areas which we're finding particularly exciting now, and some of the areas that we're looking into developing. There'll then, of course, be some time for Q&A at the end. So, moving on with this, what is the technology? Let me provide a bit of background and some basics of what we have. I'd like to start this from an R&D perspective. Those of you who may not be familiar with Regenesis, we do a lot of the product design fundamental research ourselves. Much of this is in-house. Some of it is with external research facilities, whether contract facilities or universities. And much of the work on PlumeStop was done with our own internal labs. So back in about 2007, we began to focus on the use of particulate sorbents to bind dissolved contaminants in situ. This itself is a good story, but this is not the time to go into it. We looked at surfactant-modified zeolites, we looked at organoclays, we looked at activated carbons, we looked at a range of different materials. And what we found was this: activated carbon and other sorbent particulates do not disperse in the aquifer. Granular activated carbon, for example, has particle sizes of about 1 millimeter, 1,000 microns or greater. Powdered activated carbon, the same material, even milled up small, so it looks like the powder you might have in a photocopy machine or whatever. The particle sizes are about 40 microns in diameter, but when you get them wet, they agglomerate up back up to particles of greater than 1,000 microns again. So in the image that you can see on the right of the screen, this is a sieve. It has a mesh which is bigger than the particle diameter of the powdered carbon in there, but the powdered carbon particles have agglomerated up on hydration, on getting wet, and they are no longer going through the sieve. They're just sitting there and not leaking through. This is a big deal in the formations because, well, even if they were 40 microns, there would be challenges because the soil pore throat diameter for silts and sands is in the range of 5 to 30 microns. So a 40 micron particle is not going to go through that pore throat. It's certainly not going to go through that pore throat if it's agglomerated up to a larger particle. The only way to get powdered carbons through a formation would be to fracture in place them, to go with soil parting, and this would greatly limit the contaminant contact to the edges of the small fractures or cracks or spears or whatever it would be, but not the dispersive flow that we would be seeking. So with the development of PlumeStop, the timeline was something like this. Back in 2007, we really started the work, and that continued through to about 2013. The real challenge was how we could enable the material to be transported and what we could do to, well, basically change the surface charge on the particles so that they would not agglomerate and so they would not attract and stick onto the soil surface, the soil particles so immediately. How could we do this? But more importantly, how could we do this without compromising the sorptive capacity? There were two discrete challenges, but finding the sweet spot where both challenges could be overcome was really the focus of this developmental research. We went through a range of different approaches, a range of different formulations, some of which we took to the field to test. Some of them did not work, and we would go back and work with refining the design in the lab until we hit on a formulation in late 2012, early 2013 that really was starting to show some powerful promise. The field beta tests that we did with this in the 2013-2014 period really showed that we were getting repeatedly the results that we were looking for. Those early tests are actually still running for long-term data. These are really just R&D in the field. But on the back of those tests, we gave the technology a trade name, PlumeStop, and we launched it commercially at Battelle, Monterey. The commercial application started soon afterwards, from mid-2014, and it's those which are being reviewed in this presentation. So what is the PlumeStop reagent? Well, at very essence, it's a highly dispersive, injectable sorbent and microbial growth matrix. I say sorbent and microbial growth matrix not because it's a mix of two things. Rather, it's one material, but it acts as a sorbent and a microbial growth matrix. The relevance is that the sorbent will provide a rapid drop in dissolved phase contaminant concentration and an immediate risk reduction. There's no contaminant destruction here. We're simply taking contamination out of the dissolved phase, locking it up on the sorb phase and taking it out of the mobile risk pathway in groundwater. And as the groundwater concentration drops, naturally the concentration in the vapor phase will drop, headspace analysis, as any analytical chemist will tell you. So the vapor intrusion risk and vapor-driven risk will also immediately drop. This can be monitored with a PID during the applications, and it is remarkably fast. The destruction is really undertaken, the destruction of the contaminant really follows from the growth of the bacteria on top of the sorbent material now that it is replete with the contaminants that have sorbed into it. Which bacteria are going to grow? Well, mostly the ones which are degrading the contaminant that the material has soaked up. And it's this microbial growth that will provide an accelerated biodestruction of sorbed mass. Why accelerated? Because you have a higher concentration of bacteria and contaminants in the same place, you have higher local bioavailability, you have a higher instantaneous rate of contaminant destruction. This is really one of the points that we covered in the first and second webinar, so I won't go into it in more detail than this here. It also provides an ability to clean up to much lower cleanup targets. The low micrograms per litre or sub-microgram per litre range are well within the capability of this technology. What is the reagent physically? Well, physically, it's colloidal activated carbon. The particle size is 1 to 2 microns in diameter, about the size of a red blood cell, about the size of a bacterium. This suspends as a liquid. So it really just behaves and looks more or less like an ink. It has a very large surface area, not simply because the particles are only 1 to 2 microns in size, and so naturally they'll have a high relative surface area, but because the activated carbon itself has a very high internal surface area. The surface area of a gram of activated carbon is something like 800 to 1,000 or more square meters per gram of material. And milled down fine, the immediate availability of that is much higher. But it did not take us 7 years of research to figure out that you could grind carbon up small. What we're looking at here is not PlumeStop, that's just small particles of carbon. What makes this PlumeStop was, and what took us the research time, was how we could actually prevent the particles from clumping together, how we could ensure that they were going to distribute and disperse in the formation without compromising the sorptive capacity. And achieving this was really the core innovation that created the PlumeStop product. And it's this that enables wide area, low pressure distribution through the soil matrix without clogging the formation. So actually, let me go back. We can see the image on the left there to give you an idea of the surface area that I've described with the first bullet point here. There's coming on 200 mils of PlumeStop that's being poured into that beaker on the right. There is enough carbon within that to have a surface area of about the size of a soccer field. So if you imagine the degree of partitioning that you would get of an organic material onto another organic material, and then you imagine on a molecular scale a surface area of a soccer field, that is a very, very high sorptive capacity. And that's what you can see in the beaker. So the way that we would apply it is simply this. It can be injected at low pressure, it can be effectively flowed into the subsurface at low pressure. It will disperse widely depending on the advection rate of the groundwater or the flow rate of your pumping, and we can get a wide distribution of PlumeStop from each of the application points. The PlumeStop will then coat the subsurface particles. What we can see there is a large sand particle with the little gritty bits of carbon over on the surface. I'll show you some clearer images later in this talk. Once it's in place, of course, we can stop injecting it. The material is there. We will start to get absorption of the contaminants into the carbon particles, which are now coating the sands, the silts, and the internal formation of the subsurface. Onto this, we will then start to get biofilm growth of the bacteria, which can degrade those contaminants. This will provide the accelerated biodestruction of the contaminants. So the carbon has cleaned the water, the bacteria have grown on the carbon, now the bacteria are cleaning the carbon. This then regenerates the adsorption sites so that as more contaminant mass comes into the treatment area through back diffusion or through advection from up gradient, more contaminant mass can get absorbed, and round this circle we can go again and again. The PlumeStop itself is not consumed by the process, but the contaminants that get sorbed onto the PlumeStop can then be destroyed, freeing up the sorption sites for further capture. These are some of the points that we drilled into specifically in the first and the second webinar. I won't go through the mechanisms of that in this webinar, but they are there in the earlier ones, should you wish to recap on those. To give an idea of the distribution, this is perhaps the most graphical illustration to date. These are two columns of fine to silty sands. Onto one, we're going to put powdered activated carbon. Onto one, we're going to put PlumeStop. We're then going to open the taps in the bottom of these two columns and watch what happens. This is what happens. So as your screens slowly refresh and show this, you'll notice the difference between the column on the left and the column on the right. A total of 3 pore volumes of water went through each column. But as you can see in the PlumeStop column, the carbon was simply just flowed through the column and left a coating, more or less like ink. And as an ink, the material that's left behind is stuck fast as the bead of ink continues to flow until there's no ink left. That's essentially the difference. So the difference between powdered activated carbon and PlumeStop is essentially the difference of delivery into the formation. One will block and clog and will require fracturing in if it's going to be used. PlumeStop will disperse freely, flow like water, and leave a coating. This, by the way, is what the sand particles looked like prior to the PlumeStop application. If you look at the error bar at the bottom of your screen, the scale bar at the bottom of your screen here, this is about a 50 micron particle size, a scale bar here. When the PlumeStop is on top, the scale bar is now 20 microns, you can see the coating of the PlumeStop particles on the sand. Everything looks the same color because it's a scanning electron micrograph, so it's in monochrome. But these little rough points here are going to be the black carbon particles sitting on top of the sand. This is a 20 micron scale bar, so you can work out that these are about 1 to 2 micrometers, as mentioned in the earlier slide. Another image of the same, different bit of sand, different bits of PlumeStop. Onto this will grow slowly the microbial biofilms and biomass. These are some micrographs of biomass, not on PlumeStop. We didn't have the microscopy capability to do that. There are little scale bars on these images too, if I can point them out. These are each half a micron scale bars. So if you try and do the compression in your head of 20 micron scales to half a micron scales, you can see that the bacteria are broadly the same size as the PlumeStop particles, and they'll really just grow a web of creepers and nets and all of the wonderful things that happen down in that microscopic world over the PlumeStop surface. So there are many questions that are often asked about PlumeStop. I'm going to hit on a few of them in the next section. What about distribution in low permeability zones? What about contamination in clays? What is the radius of influence? How far can you actually inject the material from one point? And many questions which are variations of those. Well, to explain this, let me use this simple image. This is really just a sketch of a cross-section through a formation, and zones that are monitoring well will see. What we've essentially got is contamination here shown in red on this sketch that is principally transporting through the higher permeability zones, the zones of principal flux, the gravels and the sandy and silty zones here, not so much in the clay in the top. There will be progressive contaminant diffusion into the lower permeability zones as the flux passes through the higher perm zones. This is what dogs the tail end of many remediations as this material back diffuses out into the water that may have been cleaned in the flux channels, and this leads to a lot of the tailing of remediation performance and a lot of the rebound of remediation performance. These are zones of residual storage of contamination outside of the flux channels. There is relatively little contamination that actually gets deep into the competent clays or bedrocks or the low permeability formations. So when PlumeStop is applied, it can flow into the higher flux channels, the higher flow channels, the higher permeability channels at low pressure, and it will flow through these, coating the surface as if it were painting them. There will be relatively, so as it leaves that coating, even though not much PlumeStop has got into the lower perm zones and none will have got deep into the competent clays, as the contaminant mass starts to back diffuse out of the lower permeability formation, it will get captured by the PlumeStop. And likewise, any mass which is advecting in from up gradient will get captured in the PlumeStop. So in this manner, the low and the high permeability zones are addressed. So this is sort of the answer to the clay question. The PlumeStop will not flow into the clay any more than water will flow into the clay, but its influence will address the contamination in the clay through the capture of back diffusing mass, etc. The PlumeStop therefore will flow like water, and ink is an easier analogy, but will leave a coating. The distance that it can move from an application point therefore depends largely on the volume that's injected. How far will a bead of ink roll over a page? Well, it depends how absorptive that page is to the ink, and it depends how much ink you used. It will keep rolling across the page, leaving an inky trail until the ink has basically all been spent. And PlumeStop rolling through a high permeability zone will be very similar. Field application then for successful PlumeStop is therefore all about ensuring accurate placement of PlumeStop into the flow zones and filling those. The key word is flux interception. The key words are flux interception. By the way, this is what these images really look like in practice. This is a soil core that's been taken out through a PlumeStop application. And within this, you can clearly see the black discoloration of the carbon in the gravelly zone here. This is completely analogous to this. In fact, this is a sketch of the same thing. Whereas the competent clays further down have none of the PlumeStop in. What's interesting, though, is that there is some discoloration here, and the PlumeStop appears to have penetrated some distance into the lower perm zone. Well, is that real or is it not real? Well, some interesting things have started to come through additional bits of work that we've been doing with Colorado State University. I'm not going to go deep into this particular piece of research. I just want to show you one of the things that we've noticed while it's been going on. But basically, this is a back diffusion project that is an extension of one of the military sector, military contaminated land development projects funded within the United States. It's been taking place in the lab of Tom Sale at Colorado State University. Kevin Sanner is the capable PhD, former student of Tom Sale, now works with CDM Smith, who's working on this. What you can see in the tank on the left is essentially an upflow column with alternate connected sections of silt and sand, and the whole idea of this was to look at back diffusion out of the lower perm into the high perm zone and how to treat it. I won't go into the detail of the experiment, but basically if you imagine that tank turned on its side, then that's the type of flow-through formation that we've created a microcosm of in the ground, so that when we apply the PlumeStop, there it is being applied through a geoprobe, and we let it flow through the formation in the direction of the groundwater, shown by that blue arrow. Then we progressively fill the high perm zones with PlumeStop, as you can see. But a remarkable amount starts to penetrate into the lower perm formation as well, as you can see here. That's basically then a model of the sketch that we looked at earlier. What I think is particularly remarkable about this is that over time the PlumeStop crept further and further through the low permeable zone, and by way of yardstick, this is how well permanganate performed in the same setting, in the same setup, with the same material. So the permanganate has progressed perhaps a centimetre into the low perm zone over the 3 months of the study, whereas the PlumeStop pretty much found its way through the silt as well as the high perm zone without getting washed out. So it is remarkably penetrative, even though it is not necessary for it to get deep into the low permeability formation. What about the usage of PlumeStop? Well, this is what a map of sites looked like at the beginning, in the middle of last month, in October 2016. PlumeStop was originally rolled out from the Midwest in the States. The Midwest is not in the middle, it's not in the West. It's this bit up around Chicago, Ohio Valley area here. And we progressively rolled out from there simply for logistical reasons. The European project started perhaps 12 months after that, maybe a little less than 12 months after that. In terms of statistics, PlumeStop has now been applied on 84 sites. Many of these had pilot studies beforehand, so there are more than 84 different applications, but there are 84 sites. They're through 27 of the US states and also US territories such as Puerto Rico. Next injection is down to Puerto Rico. I think I need to supervise myself. The number of countries that the material has been applied in is 6. So this is a lot of different regulatory regimes. There's a lot of regulatory autonomy across the states. So even though the US might look like one country from here, in terms of regulatory approaches, positions, stringencies, questions, sensitivities, it is as if it's 50 different regulatory areas, you might say. The states that the material has been applied in are these. I won't go through these. They're principally of interest to folks from the States, of less interest generally in Europe. The countries that the material has been applied in, US, Canada, Italy was the first in Europe, Belgium, UK, Sweden. In terms of these applications of the 84 sites shown, 30 of those are at the pilot stage, 54 of them are full scale. Most of those full scale ones had pilot applications ahead of them. What about the contaminants that have been treated? Well, in terms of the wider classes of aerobically degrading rather than anaerobically degrading species, it's more or less half and half. It's pretty much 40 each of sites which have had aerobically degradable species, petroleum hydrocarbons, BTEX, etc., and anaerobically degradable species such as the chlorinated ethenes, chlorinated ethanes, etc. About 4 of the sites were mixed solvents and hydrocarbons together, for example. And among the other contaminants of note, beyond the usual suspects like chlorinated ethenes, ethanes, petroleum products, BTEX, etc., were PAHs, Freon 11 refrigerants, MTBE, very good results actually for MTBE, which surprised me because its sorption is quite low, tertiary butyl alcohol, chlorobenzene. Across Europe, we began making the applications a little bit later than in the States. We learned a lot of lessons in the States before we started to bring the product over into Europe. There are 11 sites where the material has been applied to date. You can see the map on the right. There are 8 further sites which are scheduled to go over the next 4 to 5 months, actually in the same countries that we've already applied in. There are 28 further projects which are in the design and exploration phase and discussion stage at different levels of maturity and detail. And we have really found a wide interest everywhere, east to west, from Ireland to Poland, north to south, from Scandinavia down to the Mediterranean. We have experienced to date wide regulatory acceptance. I do not speak for regulators. I can only speak from our experience, and our experience to date is that although there will naturally be many questions asked by regulators, it's only appropriate, it's a new technology, there have been no territories to date in the United States or Europe or anywhere in the world where the applications have been blocked by regulators or refused. The material is designed for injection into aquifers. The components of it are food grade. We've been deliberately careful about that, as we have with the other Regenesis projects which have been applied. And there are something like 23,000 other applications of Regenesis products around the world to date. So how have these sites got on? What are the performance statistics of these? Well, earlier this year I sat down and had a comprehensive look at everything that we had done to date, all of the information that we had from as many sites as I was able to get data from. The data assessment, here's a snapshot of it, looked something like this. I'm not expecting you to read that, but essentially what we've got here are all of the data pooled from all of the available sites that I had at that time pooled, and this was from something like 24 different sites. The good, the bad, everything was pulled together. We looked at, from all of these sites, we looked at the wells which would be within the expected zone of impact on these sites. These were highlighted in the assessment sheet and assessed. So these are wells which were within the treatment grid and or a close enough effective distance to expect a result from the application on a down gradient monitoring well, for example, within the time frame that we studied. We looked at the total contaminant reductions over time and created performance histograms using the full data set, the good, the bad, and the ugly, everything. We looked at the initial capture and we looked at the stability to date. The only data screened out of this were wells in the areas which we would not expect to impact, wells up gradient of the treatment or wells too far down gradient of the treatment for the area of influence to actually reach within this frame. So this is the full data set. And the histograms look something like this. This is the reductions in concentration in the target wells over the first 1 to 3, the first 3 monitoring rounds or so. So this is typically within the first month to the first 3 months, depending on the monitoring frequency. The axis along the x-axis along the bottom shows the percentage reduction secured. How far did the concentration go down within the first 3 monitoring rounds after the PlumeStop had been applied? The vertical axis is the frequency of this. So what you can see is that the big fat bar on the right-hand side is showing that the majority of sites, concentrations were reduced by more than 96% within the first 3 monitoring rounds. Why is there no higher resolution than 96%? Why aren't I looking at 99.9% or 90%? There's a simple answer to that. detection limits were generally met somewhere around about the 97, 98 point, and so we don't actually know how much lower the concentration went than the method detection limit of that site. We simply reduced the concentration below the quantitation limit of that particular site. Up at the top end, we've got concentrations reduced such as chlorinated ethenes, chlorinated ethanes, freons, etc., of the anaerobically degrading species. We've also got TPH, MTBE, BTEX, etc., and chlorobenzene. Those are some of the things that we'd removed. Although the majority of sites were reduced by very significantly, there are these down at the bottom, which, although a small number of the sites or the actual monitoring wells that we'd expect to influence, showed no result whatsoever. And though these are a minority, they tell us some very interesting things. We looked more carefully into those, and what we found was that there's actually two of them down in that block there. One of them was a site that performed very well over the majority of the zones. In fact, the majority of the performance on that site is right up in the top histogram, part of the histogram. But down at the bottom, we found one well that really should have had a result but didn't. And when we looked more carefully at the data, we found that it was in a zone of not just made ground, but really demolition rubble. And so what we think happened there was that the material that we applied basically found a very high flux flow path, maybe a drain run or some other conduit, and simply just disappeared down that rather than filling the formation around. One of the other sites down there was a different story. It took a while to find out why we weren't getting an effect where we thought we would get an effect. It only really came down to looking at historical data of that particular site and finding that back in 2010, in the zone that the PlumeStop had been applied to, there had been an application of emulsified vegetable oils. And what we realized is that that had not been successful in getting rid of all of the contaminant mass, hence the desire to come back and apply the PlumeStop, but it had left a considerable amount of oil material in the ground, which of course the PlumeStop just stuck straight into, and it used up all of the sorptive capacity. So there is an incompatibility between emulsified vegetable oils and PlumeStop. It does not mean PlumeStop cannot be used with electron donors. It simply means that the electron donors that PlumeStop might be used with would need to be compatible. So overall in these statistics, 65% of the sites were reduced by more than 95% within the first 90 days or so, typically to below the method detection limits. 70% of the sites were reduced by more than an order of magnitude within the first 90 days. 90% of the sites were reduced by more than 80% over the same period, and only 10% of the areas that we looked at were reduced less than 65% over that period. So what about the stability? Knocking concentrations down can be done. What about rebound? What's happening over time? Well, we looked at these are the data sets from the corollary. These are really the opposite of what we were looking at before. Here we're looking at all of the data of the sites. The average period of time we're looking at here is about 200 days. The longest term one is about 738 days. And here, what we're looking at along the x-axis is how far has the site rebounded, if you like, how far have concentrations increased back from what they started from. So if anything was reading 100% up here, then the concentration has gone back to 100% of what it was before the PlumeStop was applied. Down at this end, if we've got something in the 1 to 5% here, this shows that the concentrations have gone up to 1 to 5% of whatever they were before the PlumeStop was applied, or in this case up here, 36 to 40% of what the concentrations were before the PlumeStop was applied. So 70% showed no change over the duration monitored, or the concentration went down further. That's this big bar on the left. 85% remained within 10% of the initial result. The remainder, bar 1, were pilot tests. This is significant. When we conduct a pilot test, the whole purpose of a pilot test is to calibrate the main dose that we'd need on a site. If we put an excessive amount of PlumeStop to guarantee a result, that's great for getting a good result, but it tells us nothing about whether we were exactly on dose or only just on dose or not quite on dose to get the maximum balance of dose response. So pilot tests are typically designed to be injected right on the balance of what we think is about right, so that we can then see how much more we might need to increase the dose or reduce the dose, whatever it may be for the pilot application. So it's actually a good thing on the pilot tests that we're seeing ourselves pretty much with a good initial capture, but starting to see some of the rebounds afterwards. That's a good calibration point to be at. We can see right where we are on balance. One of the pilot test ones actually was in another area of the site that had veg oil on. We still got an initial reduction, but the sorptive capacity was compromised, and so we found that we were getting more accumulation of some of the breakdown products and less capture of some of the material coming in from up gradient. Again, we'd lost some of the effective dose onto the oils that were already in the ground. Good lessons learned. So let me just take a step back from this for a moment. Here's the initial capture, and there's the stability. When I look at this, I realize that I can't think of another technology that we have available that has this type of performance record that can reduce concentrations so fast with so little rebound. I've been in the industry 25 years or more. This is really something. Thermal remediation perhaps could do this, but thermal remediation can't be injected over a wide area at low pressure. So this technology, I really do think, brings us as an industry into some new ground. I'm going to skip drilling into that any further because I want to leave more time for questions. So what are some of the lessons learned of the applications that we've had to date? Well, these are the principal ones that I would single out. Lesson number one, it's not about how much PlumeStop you can get in the ground. It's also not about how far away you can see the PlumeStop has moved. If you get PlumeStop showing up in a well 30 meters away, what does that tell you about the actual flow path of that PlumeStop? How much of the contaminant flux have you intercepted? Has it just gone down a wormhole and appeared 30 meters away and is doing nothing in between? Has it just fractured along a thin seam and is doing nothing in between? It's just hit your well and filled your well. No, it's all about how much of the flux, how much of the moving contaminated groundwater can be intercepted by the PlumeStop that has been put in the ground. The second lesson is this. Getting the PlumeStop in the ground is easy. It flows like water. It will easily disappear out of a pipe and into the ground. What is important, though, is getting it into the zones which are really going to be intercepting the flux. What we want to avoid is 90% of what we put in the ground intercepting less than 10% of the contaminant flux. This is what would happen, for example, on that building rubble site where we put a lot of material in, it disappeared down a pipe run or whatever it was and did not impact the surrounding groundwater significantly. What we're trying to achieve and what we want to achieve is ensuring that the majority of the material we put in the ground intercepts the majority of the contaminant flux. And so ensuring to how we do this then in practice, and one of the lessons that we've learned is essentially that, well, rather than sell the material, at least for the time being, we're applying PlumeStop directly ourselves using our own kit and being sure that we are targeting those flux channels. We have our equipment across the States, we have equipment across Europe for these applications. What we will typically do ahead of any of the injection work is take a good look at the aquifer itself, the main flux channels, etc., and we'll do a flux zone verification exploration ahead of the application of the PlumeStop. So what are we doing here? Well, it's essentially a pre-application field verification of the remedial design parameters. We're doing a high-resolution identification of the principal contaminant transport strata. We will typically undertake this by the design injection personnel, the people who've been involved in designing the project, so they can quickly see whether what they had designed and what they had in mind is actually what's there under the ground. And this enables accurate placement of the reagents for maximum flux interception. Why is it necessary? Isn't there already a site investigation? Well, yes, there is. But site investigations will typically focus on liability and risk assessment. Their emphasis, for example, will be on contaminant identification, plume dimensions, migration pathways, etc. The Flux Own Verification has a different set of objectives. Same area of land, same contaminants, different questions. The objective here is efficient reagent contaminant contact. So the emphasis is on the principal impacted strata, on the contaminant mass distribution within these strata, and on the parameters that we need to know for reagent delivery. So there may be water injection tests, there may be hydraulic profiling, and there'll be various forms of sorting of cores, as you can see. It's typically undertaken 4 to 6 weeks ahead of the planned application to allow time for data analysis and design refinement. But what I think is particularly striking is from a review of the data that we did earlier in this year from all of the tests that we'd done, 80% found unanticipated results. 2/3 of the preliminary designs required some degree of modification or refinement. This is not because the site investigations were wrong, they were right. It's simply that the set of questions they were asking was different. What's more striking, though, and I think very positive, is that 80% of the design changes that took place because of these were cost neutral. It was simply a matter of refocusing. So this is more to do with aiming the gun, aiming the arrow closer at the target, rather than changing the gun or changing the arrow or changing the target. This is simply a focusing exercise and a delivery exercise, and it's really all part of our strategy of successful applications. So in the last few minutes, I'd like to talk about some of the areas that we're exploring now with PlumeStop and some of the things that I think particularly exciting at the leading edge of what we're doing. Well, one of these is the integration of PlumeStop design and monitoring in due course with fate and transport models. The first exploration into this has been through using a very simple basic model from the US called BioClaw. And we've been working with Professor Arturo Keller of the University of California in Santa Barbara on developing some routines within BioClaw that enable PlumeStop doses to be put into the software to actually model the performance that we would see over time using simple modeling platforms. Because basically we can do this because PlumeStop gives us the engineering ability to place a desired retardation factor into a transport zone. Let me say that again to you modelers. We now have the engineering ability to place the desired retardation factor into the transport zones, and we can do this without changing groundwater flow. Groundwater will be unimpeded, the retardation factor will be changed. So we can use models to dial in the desired outcome and explore different design and placement options of single barriers, multiple barriers, different doses, whatever it may be. And then with a PlumeStop application, we can change this modeled retardation factor into a true retardation factor. That really brings us to the final bit I'd like to cover, and that's perfluorinated compounds. Some of you may be very familiar with this, others may be new, but these are a significant class of emerging contaminants with a number of important characteristics. One, they spread like wildfire. They move very rapidly in groundwater. Two, they're recalcitrant to degradation. They do not biodegrade, and they have not been shown to be destroyable using chemical oxidation or most of the approaches we use. Not surprising, because they're used as flame retardants. they're used in Teflon, they're used in Gore-Tex, they're used in a whole range of different things that we use. And Teflon, as you know, is not particularly good for bioremediation, as you might expect. They're also used in aqueous film-forming foams for firefighting, so they're very common pretty much anywhere where you may have an airport. There are about 6,000 of them. The two species shown, PFOS and PFOA, are perhaps the celebrities, but there are many, many of them. They're a growing class of contaminants with very, very low cleanup targets in most cases. This really shows a sorption isotherm of PFOA, one of the key indicator species of the perfluorinateds and polyfluorinated species. This is the sorption isotherm of PFOA on to PlumeStop. PFOS is similar, actually sorbs slightly more. And for those familiar with isotherms, I've put the Freundlich constants over on the right-hand side. But more importantly, if we look at the PlumeStop dose here that would be required to drop a 5 ppm plume down to a 5 ppb plume, these are the relative amounts of PlumeStop that would be required for PFOS and PFOA compared to PCE. And PCE is one of the easiest compounds to treat here. What's the take-home here? The take-home is that the capture of perfluorinateds on PlumeStop is very high. At lower concentrations, more akin to what might be treated in a PFOS or a PFOA site, then the actual capture of PFOS and PFOA is relatively much higher. In the previous slide, we were needing about half or a third of the amount of PlumeStop to capture a similar amount of PFOS or PFOA compared to PCE. At these lower concentrations, we only need about a fifth as much. So the actual capture of these species on PlumeStop is very strong. So what are these curved lines I'm showing on these graphs? Well, these are isotherms, and what they're really telling us is that, well, when perfluorinated species in groundwater encounter PlumeStop, they attach to it through adsorption and drop out of solution. The groundwater concentration drops significantly. The isotherm graph that you can see basically describes the ratio of contaminant sorbed on the y-axis to the amount of contaminant left in solution on the x-axis. They describe the partitioning equilibrium. And importantly, this ratio changes depending on the mass of contaminant in the system, hence the curve. The lower the mass loading, the lower the concentrations of the amount of contaminant you've got, the greater the proportion is sorbed. So the capture efficiency of perfluorinated species on PlumeStop is already high, but at low concentrations it gets very, very, very high. And this is a good thing. It's very helpful to us. To give an indication of that, well, another question first. What happens over time? Won't a capture barrier eventually fill up and break through, given the perfluorinateds will not biodegrade? Well, the answer is yes, it will eventually fill up and break through. What's important is how long it will take for that to happen. How long this is going to take is described by the retardation factor, and this will be governed by the contaminants, concentration, contaminant mix, and the PlumeStop dose. To give an indication of what this means, the retardation factor in modeling terms determines how fast a contaminant will move relative to the groundwater that's moving. So a retardation factor of 1 means the contaminant moves at the same rate as the groundwater. Retardation factor of 2 means the contaminant moves at half of the groundwater velocity. Retardation factor of 10 means the contaminant moves at a tenth of the groundwater velocity. For most VOCs in natural soils, retardation factor somewhere in the range of 1 to 3 would not be unusual. So let's look at PlumeStop and PFOS and PFOA. At a mid-range dose, the retardation factor of PFOA is going to be about 80. At a lower contaminant concentration, it's 570. 10 micrograms per litre, it's 4,000. With PFOS, it's even more. 1 milligram per litre, 375. 100 micrograms per litre, 2,000. 10 micrograms per litre, retardation factor of 10,000. So what this would mean then is a mid-range dose of PlumeStop, 5-meter wide barrier, 50 meter per year seepage velocity, 100 micrograms per litre of PFOS species coming in. Groundwater transit time would be something like 36, 37 days. The PFOA transit time based on a single component is going to be something like 57 years. PFOS transit time is something like 200 years. These are indications based on individual components. There will be mixture impacts, and of course some perfluorinated compounds are retarded to lesser extents, some to greater extents. That's not the point. Those are design considerations, yes, but the point is that there are some quite remarkable retardations and captures that we can get. This is at 100 micrograms per litre. At lower influent concentrations, the retardation factor quickly becomes much, much greater. So basically, PlumeStop will not eliminate or destroy perfluorinated species, but they certainly can stop a plume from spreading and prevent a plume migrating often over many kilometers and onto a range of sites around. It can keep them within a site boundary, and I think that that is at the very least a good start. So to wrap up, there are many things I've covered in this webinar, an awful lot of potential take-home messages that you may have had from it. If I can single out just one, what would it be? Well, I would say this. The first point is that we now have a technology which enables us to turn the subsurface into an activated carbon filter. We can think of it like a Brita filter in the ground. Rather than pumping water out and putting it through carbon, we can put the carbon in the ground and let the water get captured on that. The carbon will clean the water, the bacteria growing on the carbon will then clean the carbon. This will enable us to capture contaminants and focus bioremediation, enabling us to get to stringent cleanup targets of low micrograms per litre or even less than a microgram per litre much faster, and also to manage back diffusion. I should add that PlumeStop is particularly effective at low concentrations simply because of the way isotherms work. The lower the concentration, the more efficient it becomes. This is qualitatively different from many cleanup technologies which work well at moderate contaminant concentration levels but tend to have an efficiency which tails off as the concentration drops. PlumeStop is the other way around. It has moderate efficacy in medium concentration ranges, but greater and greater and greater, increasingly great efficiency as the concentration goes down. It makes it particularly useful as a tool in a treatment train approach if starting concentrations are that high. The other point is that we have a tool which can use to passively engineer plume dynamics to change the way a contaminant plume is moving or spreading without having to pump groundwater, without having to put energy into the ground. The groundwater flow will remain interrupted. Essentially, the retardation factor is now an engineering variable. So thank you very much for your attention through this time. I'd like to now pass back to Amanda to deal with any closing points and to field many of the questions that she's been scribbling down throughout this webinar. So Amanda, over to you.
Let me just see this. Yeah, how about the risk of spreading contaminants that initially soak to PlumeStop and then flow with the groundwater to another area? Can you answer that one?
So I guess the question is, there seems to be a paradox here. If PlumeStop moves so well and sorbs contaminant mass, why isn't it going to sorb contaminant mass and continually moving and continue to move well and then carry the contaminant off-site? It's an important question. The answer is this. The first point is that the PlumeStop will only be mobile for a period of time. The treatment that secures the movement of the material will last for about 1 to 3 months, depending on the setting. After that, all of the material will be just locked in place. Ahead of that time, the material will leave a coating really from day one, and that coating will not wash out. We've done tests in the lab which show this, but to me, the most convincing indication of just how well PlumeStop will stick to a formation from day 0 is from when the material has actually surfaced during application. We try to avoid that, but sometimes it happens. Then the black carbon that's been spilt on asphalt from the PlumeStop will not come off with a jet wash on day 1. So the PlumeStop that's stuck onto the soil is not going to move. If you imagine the PlumeStop moving through the formation and being mobile for the 1 to 3 months, the trail that it leaves will be stuck. The moving front will continue to move, so we have to look at how far the groundwater will move within 1 to 3 months. Generally, it's not a problem. It simply extends the radius of influence. But if there is a sensitive receptor, property boundary, river, lake, or whatever it may be within an advection distance, then it is possible to lock the PlumeStop in place there and then through injection of a counter reagent, which actually provides a means of dropping the mobile PlumeStop out of solution in a flash. And that's one of the things that we can do with our field application capability. So the PlumeStop will, again, I use the ink analogy. The bead of ink will roll over a page until there's no ink left, and whatever's stuck to the page is stuck and will stay stuck. And so that's why the contamination is not going to be transported by it.
Okay, thank you, Jeremy. Again, I've grouped similar questions together. Let's see. Yeah, how well will it perform in fissured chalk or fractured rock?
Okay, fractured settings. A lot will depend on the type of fracture that you've got. If they're large block fractures or saprolites, then it goes in pretty easily, and we've had good results of, I think, a couple of sites that we've done to date like that. Hairline fractures, granites, etc. I would look at these carefully because the material does remain particulate and you wouldn't want to block them. Chalk or dual porosity systems, again, we've not done any yet, but the movement should be pretty good. The capture of PlumeStop on these materials should be reasonably strong. They're not precluded, but they are things that we need to look at carefully, case by case.
Thank you. Right. Another question here. You mentioned modeling and the use of the adjusted BioClaw model. Can other models be used, such as the RTM P20, ConSim, or ModFlow MT3D?
The answer is yes. A wide range of models could be used. If that's probably too much to get into in this answer, but if modeling is your thing, and if you're interested in how to dial in PlumeStop parameters into any off-the-shelf model, then please contact me, and we're best dealing with that externally to this call.
Okay. I think we'll have time for about one or two more questions, perhaps.
Yeah.
Right. Another question is, won't the carbon itself or the bacterial growth block up the aquifer?
So we're putting carbon into the ground. Could we be, when it's particulate, could we be blocking flow channels? And all of that microbial growth, could that foul up the aquifer? Well, let me deal with those one by one. Well, the overall answer is no. Let me qualify that answer and explain it. The blocking of a formation through microbial growth typically will not occur unless we're looking at a point source of microbial growth, say around an injection well that's putting in a high bioavailability electron donor such as molasses or lactate or whatever it may be. Dispersed into the formation, the microbial growth typically does not change the permeability of a formation, as we know from all of the ORC and HRC and other projects that we've done over the last 23 years or so. What about the carbon? Well, the amount of carbon that PlumeStop actually leaves in a formation per unit mass is actually quite small. So the FOC of PlumeStop carbon, it's not organic carbon, but the FOC would be about 0.001005. So we might actually be doing something like doubling the FOC in the formation, which clearly isn't going to block it. But because of the extra sorptive capacity of the PlumeStop, the effective FOC, the actual sorptive capacity we get of that tiny change, is going to be something like 500 times greater. It's going to depend on the contaminant and the concentration and the isotherms and all of that, but we might actually change the FOC by 0.1%, but we might get an effective change in the FOC in terms of retardation of 1,000-fold for that. So we're not going to block up or clog. Would PlumeStop work for chromium-6? Actually, yes, it would. We haven't tried it with chromium-6. Chromium-6 can be treated on activated carbon, and there are probably many people listening who've used activated carbon pump and treat systems for chromium. Whether it's the best approach is another question, but theoretically it can be used. I would compare it to a number of other different treatment technologies, but the answer is yes, it can be used for Chrome-6. What have we got here? Amanda's pointing to another question. What are the typical dissolved phase TPH concentration ranges that PlumeStop will treat? Okay, when to use PlumeStop and when not to use it. It's not a technology for free product. Let me say that again. It's not a technology for free product. The typical range of concentrations that we would look at the use of PlumeStop for would be below 10 milligrams per litre, and as you get down to 1 milligram per litre and below, it becomes even more efficient. So yes, we've treated sites at 20 ppm and so on, but around about 10 and below is typically the range that we'd look at. With hydrocarbon sites, beware of overlooked mass. They can become a problem as well, excess sorbed mass. So 10 ppm and below would be the target zones, and 1 ppm and below it'll become increasingly efficient. 100 micrograms per litre and below it's scorchingly efficient. And I think we have probably one more question. Amanda's passed this to me to read. Will plant root growth reduce the distribution and performance of PlumeStop? Actually, yes, it will. And we know this from one of the beta tests that we did on the Central Coast in California, where there was a tree between one of the monitoring wells and the application wells. And yes, the preferential flow paths that the tree roots can put in place can mess up the distribution. So things like that need to be looked at. However, in many cases where the technology is going to be used, it's going to be well below the level of ground that actual tree roots or material or plant growth is going to be in. And I think we are over our hour now. So Amanda, let me pass back to you to close. And thank you everybody for tuning into our webinar.
Yes, thank you, Jeremy. This concludes our presentation for today. I'm afraid this is all we have time for within an hour. Thank you, Jeremy, and thank you to all who attended today. I hope you found the time well spent. Have a lovely day. Goodbye.